Student, now we want to learn a next pteridophyte. The pteridophyte we are going to learning now, that is a Marselea. The Marselea is a kind of fern. First we will see the classification of Marselea. Marselea belongs in division Pteridophyta, class Phyllocophyta, order Marseliales, family Marseliaceae, and genera Marselea. Commonly, Marselea is called as a water fern. Then we will see the external morphology of Marselea. There are about 65 species of Marselea distributed all over the world, more commonly found in tropical region like Africa, Australia. 6 species of Marselea occurs in United States. The plant body of Marselea is like a small bush. It is differentiated into the underground horizontal creeping rhizome, roots and leaves. The slender creeping rhizome is branched. It may either grow in water or attached by roots in the damp soil. This underground branched rhizome or rhizomatous stem bears nodes and internodes. From the node towards the upper side, it produces the leaves, and towards the lower side, it produces the roots. The adventitious roots grows downward and leaves grows upward. If you will see a figure, you will see some of the leaves are in a coiled condition. That is a common characteristic feature of the fern pteridophytes, and that coiled nature of leaf is considered as a circinately coiled. That is called as a circinately coiled leaf. The stem or rhizome either creeps or remain attached to the soil. The leaves are with a long petiole and compound in nature. There are 4 leaflets are present in the leaf. Each leaflet is obovate. The venation of the leaflet is dichotomous. You can see in the figure. You will see the veins are having a branches, and that is in a dichotomous manner. We want to see the anatomy of the different organs of Marselea. That is anatomy of rhizome, anatomy of root, and anatomy of leaflet and petiole. The figure you are looking, that is a TS of leaflet of Marselea. The leaf of Marselea having a long petiole, and at the tip of that long petiole you will see the 4 leaflet or pinnae are present. The leaf is compound. You can study the anatomy of leaflet by taking a transverse section and stain by a double stain combination or single stain. The section shows the upper and lower epidermis. In between these upper and lower epidermis, you will see the mesophyll tissue are present, and in the mesophyll tissue a vascular bundle is present. The stomata are present on the upper as well as lower side. In case of aquatic species, the stomata is present more on the upper surface of the leaf. The mesophyll of leaf is differentiated into palisade and spongy parenchyma. Just above the upper epidermis you will see the compactly arranged columnar types of cells, and that are the palisade tissue. So that is a palisade layer of mesophyll tissue. Below this palisade layer you will see the storage region or spongy region of the leaf, that may also called as a spongy parenchyma. This spongy parenchyma is present below the lower epidermis. You will see the large number of air spaces are developed, and the cells are spherical. There are many vascular bundles are present in the leaflet. Each bundle is concentric. At the center there is a xylem, and that xylem is found surrounded by a phloem. The vascular bundle shows clearly visible pericycle and endodermis with each bundle. Now we will see the anatomy of the petiole of leaf, that can also be studied by taking a transverse section. The outline of section is circular. There is a outermost single cell thick layer, that is a epidermis. Below epidermis there may be a one or two layer of hypodermis is present sometime. The epidermis is without cuticle. The cortex of petiole is differentiated into an outer and inner zone. The outer zone cortex has many air chambers and separated by a narrow radially arranged parenchymatous cell, whereas the inner cortex has a spherical parenchymatous cells. They contain starch, and also some tannin cells are seen visible. The inner cortex is also multilayer, but that is without air spaces. The stele of petiole is a protostele. The single layer of endodermis followed by a pericycle. The xylem of petiole is a V shape with exarch protoxylem. There are two arms of V are slightly curved and separate. Each arm has generally one or two larger tracheids in the middle and smaller tracheids towards the both end. The open end of V always point towards the adaxial side of the petiole. The xylem is surrounded by phloem. The special interest in the anatomy of petiole is shows hydrophytic character, that is presence of large number of parenchyma in the outer cortex and presence of V-shaped xylem. You can also see the diagram visual to you. That is what a TS of petiole of Marselea leaf. In this figure you can see a dichotomous venation of leaflets. You can see the figure, that is a TS of rhizome. The outline of section is circular. This section from periphery or outside to center shows the different layers of tissue, that are epidermis, cortex and stele. The epidermis is the outermost single cell thick layer and without stomata. That indicates the epidermis in case of aquatic plant, that also lacks the cuticle. The cortex of rhizome differentiated into three zones, that is outer region, middle region and inner region. The cortex of outer region is with a parenchyma. There are air spaces you will see, that is multi-layered, whereas the cortex of middle layer is sclerenchymatous, and again the inner cortex is parenchymatous but without air spaces. The delimiting layer of cortex is developed, that is a endodermis, single cell thick, and surrounds the stele of a rhizome. Next to endodermis there is a single layer of pericycle, and inside there are xylem and phloems are present. Here the xylem is amphiploic, means they have a outer layer of phloem and also the inner layer of phloem. Outside the xylem there is a presence of phloem, and inside the xylem also there is a phloem, and that is what called as amphiploic stele. The xylem is in between outer and inner phloem, and inside there is a sclerenchymatous pith. This is what the anatomy of the rhizome of Marselea. Now we will see anatomy of root. The section of root also shows circular outline. The outermost layer is epidermis, that is a single layer. The cortex in case of root is also differentiated as a outer and inner cortex. Outer cortex has many air chambers and separated by radial septa. The inner cortex has either all parenchymatous cell or some of the cells towards inner side becomes a thick walled and sclerenchymatous. You will see the two layers of cell formation in the inner cortex, parenchymatous and sclerenchymatous inner cortex. The vascular bundles found surrounded by a single layer of endodermis, followed by single layer of pericycle, and inside you will see the phloem seen. Phloem surrounds the xylem. Xylem is at the center, that is diarch in nature, and exarch condition is there, and that xylem is surrounded by phloem cells. The protoxylem is facing towards the periphery, and metaxylem are at the center. The special feature of anatomy of root is there are presence of parenchyma. In this way, in this video we seen the morphological characteristic feature of the Marselea and anatomical tissue differentiation in the different organs of Marselea, that is petiole, leaflet, rhizome, root. These organ anatomically also we studied.